Hello, welcome back. I hope everyone's doing well. We're getting down to the nitty gritties. I'll take a few moments here before we get moving on the lab. Those that I have the class and lab with, I had already sent around a revised date, a calendar for the various events, and the one coming up is the lab practical. And it will be online, of course, and it'll be 60 questions, multiple choice. And if you've had a chance to take that preliminary exam or the practice exam, however I named it, it gives you a feel for how the exam will go. I mean, the logistics. It seemed like the feedback I got, everyone seemed to like it fine. I'm allowed to include pictures and various things and reports back and that sort of thing. And what I wanted to do is I provided a lab practical study guide, and you can go through that. This voiceover might help just in general terms, but a lot of it can be done just by reading through the material and reviewing the best we can. Now remember, this is best efforts. I understand this is it's been really difficult for me. I don't like personally doing labs in biology, microbiology online. I didn't sign up for that, but that's okay. We're all called to do things at times that we have to do, and that's fine. We do it, and there's no reason why we can't have fun doing it best we can. So we're going to do that. I have sent around an announcement for a review on Lab Practical 2, and I'll do this under Zoom. I don't like Zoom, but it seems to handle larger numbers better than Teams does. It may just be checking your emails. It may roll to Teams, I don't know. There are some concerns with Zoom and security, but for us older adults, I mean, we are adults. I've not experienced any of the issues, and so far it seems to be working well. I do have a bandwidth problem. I use it for office hours, and depending on the day, Spectrum decides whenever the bandwidth is enough or not. I don't, and sometimes Zoom doesn't work, and I try Teams, same thing, and it's bandwidth. And I've called them. I pay a lot of money for extreme speeds and all this. My wife does teach as well. She is high up in Wake County Public Schools, and heaven forbid if I take any of the bandwidth for that, but I'm just kidding. There is a competition just within the home here. It's one of the limits. So we have to work within what speeds that we have. So anyhow, then the lab practical, the last day of classes is the next week, and I wanted to position that the timing here. Now, the last day of classes, of course, is on the 8th. So we'll have exam 3. Now I will do the same thing for review for exam 3 at some period of time in the early part of the week, Monday or something like that, and help you with that, but we'll worry about that later. Now for those who just have lab with me, obviously you don't have to worry about that. You get with your instructor and the timing. So all of this down here, as far as your exam, final exams and exam 3s, you get with your instructor. But what is true is the last day of classes is on the 8th, and 11th through the 15th is the final exams week. And I have chosen, at least in this block area. Now if there's other times, what this means, these lines mean, is that I leave the exam open, so any time during these days you can take that exam, and it could be at midnight, it could be 2 o'clock or 3 o'clock in the morning for all that matter. That's fine. You'll have an hour and a half. I haven't decided. It's 60 questions, as I mentioned, and you can do that, and then you'll know right away what your grade is. I'm going to set it so that you know immediately what the feedback is. Speaking about grades, I will be sending those out on Saturday or Sunday. Grading has been a huge effort. I have to intersperse that to writing and developing some of the things that I'm sending out. It does take a huge amount of time. I have to be honest about it. I'm not complaining. I'm just saying it just takes time. And one of these days I'll visit my dog and see what's going on otherwise. But if that's good with you, this is the schedule. Love to get your feedback. I'll be flexible on these times. If for some reason that within the confines that I've provided here, it's not possible for you, don't worry. I will work it out for you specifically. And that doesn't mean that I need 180 specific individual times for everybody. I have a lot of students, but I will do whatever I can to accommodate. I want you to do well and feel like you got something about it. And that's the number one thing. I want you to finish leaving this course that, gee, you know, I learned a few things. Instead of, well, now I'm done, I'm going to forget everything. I hope it's been a process that that you like and want to retain things. And so, anyhow, lab biology 110. We're going to go and cover Unit 8 on microorganisms, and that is in your lab manual that I provided for you. All of this has been posted. We tend to think about bacteria and microorganisms as things that create issues, problems, diseases for us. And through the years, I mean, this is no joke. Some of these diseases have killed millions of people. The tuberculosis outbreaks, devastating. We're talking millions of people. Cholera, anthrax, you name it. We've gone through it, and now we're experiencing pandemics with viruses. It seems like every 100 years we get some sort of nastiness. In 1819 there was the outbreak. In 1917, I guess it was, or 1911, somewhere around that time frame, we had yet another major plague that killed millions of people. And here we are with SARS-2, the particular nasty COVID-19 virus. And I'm tired of hearing about it. I don't even want to read about it anymore. And I'm a microbiologist, believe it or not. I've reached my saturation. I've gotten to the point where I don't even know what's true and what's not true at this point. And I can imagine your frustration. But hang in there. Truth always makes it avail at some point in time. So we'll address some of that today. The majority of microorganisms, they do make a lot of contributions to our well-being, the planet in general. And what I mean is that they're important for the food, sort of breakdown of food, the chains and the various things. They can improve things if we so desire. We have just sent some papers to some of my friends that there's plastic eating microbes. And can you imagine, I don't know if you've seen the huge amount of plastic waste floating around in our oceans. It is, I'm embarrassed as a human being to know that we can be so irresponsible. I had no idea how bad the problem is, and the impact it's going to have on wildlife, sea life, is, I don't know. But we need to do something about it, spend some money to clean up our act. And, you know, I'm not a really big proponent on certain things, but that is irresponsible on our part. Maybe these bacteria that can eat the plastics, break it down to less toxic type of materials and easier on other life forms. Maybe that will work. That's called bioremediation. I'm really excited about that field of bioremediation. If anybody wants to do an extra credit report, choose some form of bioremediation. And if you can read my handwriting, bioremediation, that's an important topic. I will give you extra credit for that. Don't spend much time. I know you've got lots going on. But my guess is, and I've already had some feedback, some students really like that topic. And I think that's an area of genetic engineering that can be really exploited for really good things. So I'd love to hear from you. The other topic I'm really passionate about is probiotics. I'm going to mention it here, but these are the microorganisms that colonize us, and mostly in our intestine. Oh no, I don't want you to get upset, but they are right now growing and crawling all over every aspect of your body, and your skin, underarms and feet, other places. Just it's unimaginable, but they're there. It's actually part of our defense system, if you can believe that. It's truly remarkable. So probiotics is something else that you can actually take probiotics and improve your health. Check with your doctor first, always, but they're important. So the commercial benefits, that they're used to make certain organic acids and enzymes. Believe it or not, bacteria could be used for laundry detergent, and some of the enzymes they produced are isolated and provided as laundry detergent. And a lot of companies that do that are right here in Research Triangle Park. And so now a lot of folks aren't aware of that. The other is the food industry. Vinegar, pickles, sauerkraut, beer, wine, soy sauce, buttermilk, cheese, yogurt, bread. Gluten-free bread is one of my sore points. I can't handle gluten. I'm not even an IBS or irritable bowel patient or anything like that, but I know my joints hurt and other issues I have with inflammation are exaggerated by gluten. And gluten are wheat type products. The gluten isn't the same as the gluten that I had when I was growing up. It's changed chemically, and it's been changed for more manufacturability and that sort of thing, but it does have an impact on your health. And you can have a bread, if you like most gluten-free breads. It's not the same. It is a rice, and I've gotten familiar with it, and I'll eat it if I have to. But sourdough bread, oddly enough, is a product of bacteria, and the bacteria eat the gluten, and so essentially it is gluten-free bread, and it's a product. And a lot of folks aren't aware of that, and I learned that when I was in food micro at State. Took a course. It was a great course. I would highly recommend anybody take a food microbiology course. It's just fascinating to me, and I think you'd find it the same. But gluten-free, if you want the best real tasting type of breads and things, I might point you to the sourdough bread, and you might have a good experience with that. So only a small percentage of bacteria that we have on this planet are actually responsible for disease. Now, I'll just tell you, my point of view of disease is that the organisms aren't created or spontaneously form, or however you want to look at it, to cause mayhem and death. All they're doing is growing in a niche, and that niche is their area to get food and supplies and things. If that niche happens to be you, so be it. And they will. And even if you are in good health, there's microorganisms that can take advantage of you. If you have a suppressed immune system or some other disease going on, then you're highly vulnerable to other types of bacteria and viruses and various things that we're going to try to grow and utilize this niche. If that happens to hurt you, then they don't care. It's their niche. They're just trying to grow in it. That's it. That's my philosophy on that. So we call those microorganisms pathogens or pathogenic. Bringing forth, that's ick means an unusual condition. A gen means bringing forth. Path means illness or sickness of some kind, so they bring forth. They can cause disease, but that's not their sole goal. Their sole goal is just to live and survive, as I've already mentioned. So the pathogenic bacteria belong to this group called prokaryotes. We've already talked about this in class, and I'm sure those that haven't had me for class have talked about it as well. Prokaryotes lack a true nucleus. Now, they have nucleus, per se, is this huge round piece of double-stranded DNA. And I always bring in a rubber band in the class to show you. It's the biggest rubber band I've ever seen. It's huge. And it sort of represents the chromosome, a big circular piece, and attaches to the membrane when it undergoes replication. And when we talk about that, they just binary fission, they just separate. And then they put one copy of the chromosome in one section and the other by its attachment to the cell membrane. And it goes on for that. But eukaryotes have specialized rooms or areas for the nucleus. So it has, the nucleus has this double layered phospholipid bilayer, and it contains and protects, as we've talked about in class, the DNA. The prokaryotes are sort of like a big bathtub, and all the pieces and parts are in the bathtub, as I alluded to before. Prokaryotes lack, that double membrane, and they do possess ribosomes and most of the machinery that we see in eukaryotes, but it's different. It's smaller in size. There's 70S, not 80S, and those sorts of things that we've already talked about, but it's not important for the lab. But we take advantage of those differences as we're going to talk about antibiotics and things like that. So we have to understand huge basic differences between bacteria and our cells, that leads to ways to try to deal with these pathogens. Most bacteria possess a cell wall. Now, the reason why they say most, there are certain types of bacteria some really bad, nasty pathogens like tuberculosis, or we have other obligate intracellular parasites. Obviously, if they're intracellular, they're not going to need the armor that they normally have of these cell walls. So some of them don't, but most of them, if not all, are pathogens. Some produce this capsule, this really snotty material that covers the outer part of the outside of the bacteria. Now, you can see it on the plate. It looks like someone sneezed on the bacteria plate. We grow bacteria on petri dishes with agar, and we supplement the agar with hamburgers and cheeseburgers, and, well, food that the bacteria like, which are amino acids and various things like brain heart infusion. Sounds yummy. Anyhow, bacteria love to eat that, and we get them to grow, and they produce a colony. A single cell will grow and grow and grow until they pile up to a point where we see it as a colony. And these sometimes you can take a loop, a microbiology loop, touch it, it strings. It's really snotty. It's nasty. Anyhow, most of the ones that do this are trying to evade the immune system, and it's trying to do this by tricking our immune system into thinking that our immune system is busy doing surveillance and sorts of things. And there's this thing, excuse me, about self and non-self. And some of these microorganisms produce a polysaccharide coating. Now, this is, to an untrained eye, I guess they look almost the same. But these are bacteria that don't have a capsule. This one does. You can see the residue. See, this is done under a scanning electron microscope, and it dehydrated the sample. And the polysaccharide obviously is coated all the way around. It's just, if you can imagine blowing your nose and having snot all the way around, I know that sounds gross, or bacteria, that's usually what it is. And what we're talking about with the polysaccharide is that it's a repetitive sugar, sort of like this. Now, one of the poorest things for our immune system is a repetitive type of thing like sugar. And polysaccharides are that, but we also have this self and non-self. So we have self, where the immune system says, all right, immune system, do not raise an immune response against anything that's existing in this body at a particular time in our development. After that time, then that is non-self, and the immune response can happen. So the differences between these two are really, really important. There are diseases, this autoimmune diseases and things, that's actually because of this. The slights are different. So polysaccharides, the snottiness, are made by these repetitive sugar type of deals. And we can see it without using an electron microscope by simply putting a simple acid dye, or actually a positive charge dye, and stain the bacteria, and you'll see the around where that polysaccharide is will not stain. And that's because the polysaccharide doesn't allow the dye to stick to it, and we can actually see the bacteria. And then this clearing zone, which is representing of a capsule, which is just really the repetitiveness of sugars that makes that slimy material. Now, you Star Trekkers, that's like a cloaking device as far as the immune system is concerned. So that's what's going on with capsules and pneumonia. And in the respiratory system, if you get a lot of bacteria that are growing that produce a lot of this a way to evade the immune system with this polysaccharide, Klebsiella pneumoniae does that. There are several types of organisms that will do it, and they're sticky, and it's just all part of what they are trying to do and growing in that environment. And obviously they love oxygen too, so we try to use drugs to combat pathogens, to get rid of them. You already aware of antibiotics, anti-life substances, and a lot of these are from bacteria, some of them are fungi and other forms of life that are competitive amongst one another. That's one of the areas too I really enjoy, is looking how organisms of different species interact. It's very, very interesting, but one of them is to try to exclude others. Bacteria will do it by secreting all sorts of nasty chemicals that is not so much harmful to them, but it is to others, and it's one of the ways they do that. So we have to understand the differences, and I have a little slide here looking at some of the bacterial shapes. I do want you to know the basic shapes, and from the standpoint, it's important to know these. If you were to look at these under a microscope, which is really easy to do, I wish I had spent more time. You would have had more scope time in this particular lab, but it is what it is. But what you look for are these basic shapes. Now, I made drawings here. If these coccus are arranged in chains, then that's a streptococcus. And of course, we use Latin and Greek for sciences, especially in the medical and health sciences, but it's systematic. And I always recommend my students take a medical terminology course so you can help speak the language, and it helps you also for pronunciation, that sort of thing. But chains or strep, grape-like clusters will show up under a microscope if you look at and that's Staphylococcus. And that's just based on the way they divide and all that sort of stuff. But when they do, they leave a characteristic trail behind, and that's one of the ways we can try to identify these. Now, they're trying to look at maybe a bacillus or a rod shape, as we see in the book. Sometimes we get these really short, blunt rods, and they're small, and are they cocci, or are they bacillus? So we call it either a blunt rod or a coccibacillus. And so it's that transitional shape, but we see those. And there are some look like a comma, like Vibrio cholera. The shape, when you look under a microscope, might bend like that. They kind of look like commas. We call them vibrios, and there's several microorganisms that look like that. And then we have the spirillum and spirochetes, and these come in various different forms. I can give you an example. A spirochete would be the Treponema pallidum. Treponema causes syphilis. It's really nasty venereal disease. And what's odd about these is the outer membrane. If you were to look at the outer membrane, it spins, it rotates, but the center part of the bacterium doesn't move. It's almost like a mechanical auger that drills right in. In fact, that's what it does. When it gets into the skin, it kind of just spins and drills itself or augers itself right into the membrane. Anyhow, there are others that have these basic shapes. We have the Borrelia burgdorferi that causes Lyme disease, same sort of thing. It is shaped like this. So we have a lot of important pathogens, bacillus like E. coli. E. coli could be an intestinal pathogen, or salmonella, typhi or typhimurium, all sorts of different strains of bacillus. But those are the basic shapes, and please be able to recognize those. The other way we have to talk about is how we deal with these bacterial infections, and we use antibiotics. And we want to know if an antibiotic sensitivity test is effective for antibiotics. We have ways to do this, and one of the activities I wanted you to look at was looking at using these antibiotic discs and placing them on a lawn of bacteria, and you allow the plates to grow that are going to be eating the nutrients in the plate, and the discs are really just simple Whatman blotting paper that's been soaked with a particular antibiotic. And you can see that I've got 1, 2, 3, 4, 5, 6 different antibiotics, and they're all representing, and you place them on a lawn of bacteria. All you do is you take a swab and you kind of swab the deck of the plate with the bacteria that you want to test, usually from somebody who has a disease, and you want to figure out which of these antibiotics that you want to treat the patient with, and to try to rid them of the disease. Now, this is a really important thing to do, and it's called the Kirby-Bauer method, and it goes through protocols and standard operating procedures and blah blah blah. But what it is a systematic. We actually measure the distance across the zone of inhibition, that clearing zone that we see with these. Here's one that doesn't have one, and you can see the zone of inhibition. In other words, the bacteria does not grow in those zones. And that's what we're after, is if they don't grow next to that antibiotic. You see, on the disc, it starts to permeate that antibiotic into the media and diffuses outward. Everyone knows what diffusion means, and it slowly diffuses. Well, there's a concentration at which the bacteria say, "Oh, I can't grow in there. There's just too much of this stuff," and it kills them or does whatever the antibiotic does. And it's a way for us to screen the bacteria that are causing an infection with antibiotics to see which one might be useful in treatment. And so if we use neomycin, neomycin will actually kill the microorganism, and it seems to be pretty effective. We can measure the width of this, and you plug it into a formula that's well established. If you've grown this plate overnight using the Mueller-Hinton media, blah blah blah, and you get all the right things done, that measurement will tell the doctor how much of the antibiotic to give and how often. So you may give 30 micrograms of neomycin BID, or twice a day, and the formula will do that for you. Instead of guessing, it actually has been worked out with various parameters. So I wanted you to at least be aware. Now, they've automated this to some degree that doesn't require you to actually make the plates anymore like this, but it's okay. It's the same. The concept's the same. So that is the antibiotic sensitivity test, and all I want you to do is think about which of these would you want to use. Now, obviously, like F is right here is erythromycin, and for whatever reason, erythromycin, the bacteria grew right up to it. The bacteria is resistant to it. Obviously, you're not going to give that antibiotic. It does nothing. It would do nothing. It's predicted here. The bacteria just ignore it, and so you wouldn't use that particular one in a treatment. So I wanted you to look at that, and then through that section there, you can make your determinations and fill that out, and then answer some of the questions about that. and that it's an important, I would say most of you, if not all, have had an antibiotic at one time. Now, these antibiotics, the mode of action and how they do certain things are key, of course, to the antibiotic. And what you want is an antibiotic that goes after the bacteria and not you. And so that's why we study the differences between prokaryotes and eukaryotes, is to understand those subtle differences, and then we try to design, develop, however, find antibiotics and things that kill the bacteria but doesn't hurt us. And that would be an ideal thing. And would an antibacterial agent usually be ineffective against a fungi? Now, that's fungi, not fungi. Fungi is somebody you invite to a party. Fungi is a microorganism that's pretty resilient. It resembles bacteria to some degree, but it is rather significantly different. And I can show you at least here. If you look at bacteria under a slide, those basic shapes that I showed you before, the fungal infections are different. The yeast cells kind of look like these little budding sorts of things. They have nodules, and they have filaments and various things. These are quite different from bacteria, and they are much more difficult to deal with. We actually have three basic antifungals that we use and have used for a long time. It's a quite different organism. The myconozoles and all the different types of antifungals, we're finding out now those are becoming resistant to the typical antifungals like myconazole. That's pretty scary. Treating and dealing with fungus is hard to do. They are fundamentally different. They're higher levels of organisms, and it's harder to treat these. And so a lot of the things that we do with antibacterial agents wouldn't apply to the fungus because they are different. But a lot of times we use antibiotics to try to go after certain differences that we have from our cells. The closer you get to eukaryotic cells, the less the choices are, like fungus, to stop them. So we grow these bacteria and various things on plates, and here's a representative one here. I wanted you to go around to sample, just randomly sample maybe a toilet seat or a doorknob, or the surface of your phones are nasty, let me tell you. We get sorts of bacteria like these off of phones. There'll be Staph aureus on here, Micrococcus luteus, all sorts of different types. Look at the colors and colony types, and these are all representative of bacteria that are different from one another. Most just by looking, I don't know for a fact, but that's what it appears. There's some red ones and some orange and yellow and white, and it's all sorts off of a cell phone now. The only thing I've seen worse than cell phones in practice, and it isn't toilet seats, and it's off of shopping carts in grocery stores, the handles. I don't know who touches those things. I don't know if they're from this planet or what, but there were things on there I could not identify. In fact, I took it to a diagnostic lab. I was serious about it. I wanted to know what it was. They didn't know what it was. It was Rollins Diagnostic Lab. That's what they're there for. And I didn't have the money to pursue it as new species, but I don't know if I really wanted to at the time. What we want to do is to be able to stain the bacteria and to see them under a microscope, and that would be really a useful thing to do if we want to look at the bacteria under a microscope and to determine their basic shapes and that sort of thing. And here's just some of the basic shapes, and we need a stain, what they call a vital stain. Vital stain just means that it sticks to the bacteria. One of the things I wanted to point out that's important, it's in this section here, and every now and then I stick in YouTubes that will help you go through and understand perhaps better than I have A lot of times I don't have the graphic. You can see how high and sophisticated my graphics abilities are, and I actually find the good old pen and paper is about the best that I can do to convey. And see, I can even do animations. Well, that's not a useful one, but the idea is I can hold my models and various things using this kind of forum, and I'll leave it to you to let me know if that's a useful one or not. Anyhow, so gram stain, I have a YouTube video. It's embedded in the study guide, and you can go and hit those YouTube videos. It is part of sort of the understanding what's going on, and I think YouTubes are very helpful. I try to make the YouTubes myself because they're the size are smaller, and it just, you can stream them, and it's better than an MP4 and having to download these huge files and all that stuff. Anyhow, lest I digress, it just helps in the communication. So be looking for those, the YouTubes, and they'll help you if you want to view them. Gram stain, we use a crystal violet dye which binds to a part of the cell, and I think I have, yeah, I do. I have a diagram here that may show the differences in, if you look at sort of the difference between a gram positive, will have this much thicker outer cell wall, and then it has the phospholipid bilayers under that, and that's about the best animation that you're going to get from me. Unfortunately, I can't draw a stick figure, but the idea behind that, this is your phospholipid bilayer, and that's the membrane, but on the top of it we have this really thick cell wall that's made of substances that the crystal violet can actually bind into. Now, I don't know, the gentleman that came up with this stain, Gram, and we honor that by naming it the Gram stain, why crystal violet can bind into this peptidoglycan layer here, and it wedges itself in there. And then he came back, which is really kind of blows my mind, with a substance called iodine, which we've used in the lab. It stains potatoes dark purple. If you remember, we used the iodine to identify certain types of starchiness, starches and things. But what it does is it binds and locks in the the peptidoglycan layer, this crystal violet. And when it does so, it makes it really, really tough, and it stays in it. They call it a mordant. And if you're familiar with bricklaying, they use that cement, and they call that cement a mordant, and that holds the bricks into place. Well, this iodine holds the crystal violet into place. And then we come back and we try to get or remove that stain with an alcohol. It's a Gram alcohol. It's an acid alcohol. And if it can get rid of it, it will. And it destains the bacteria. It won't have any color unless it's a Gram positive, and that particular stain gets embedded into that peptidoglycan layer, and it doesn't come out. And then we come back. Now, the ones that the stain does get washed away, we have to stain the bacteria so we can see it. So we call this a counter stain, and it's saffron. And the saffron is a pinkish color. It's a light red or a faint red. And so the purple, which is Gram positive, so positive and purple, if you can equate those two, is always the dark blue. And then the Gram negatives are this reddish color. And the Gram positives are purple. And remember that. But it's a classic, and we use this in medicine all the time. You want to do a really quick stain. Now, I've worked in a diagnostic lab. We have to figure out what to give patients. And so what you want to do is immediately do a Gram stain and determine, well, if it's a Gram positive, then you can use these broad spectrum antibiotics at least to get the treatment started, and we know they're effective against those. Or if it's a Gram negative, we know what antibiotics to give. that are active against gram-negative. So it gives us that sort of background to be able to do that. And I already mentioned, then we check the zone of inhibition, and if the bacteria are resistant or sensitive to a particular antibiotic. And the write-up in here tells you all those procedures. I don't think there's any really need to go over that, and then look through the questions here. And then now I want to move to disease transmission. And you can imagine if we go around and contaminate plates, and not plates, but handles on grocery shopping carts, or doorknobs, or the handles that get into the toilet areas, and you remember there was studies that it's kind of gross, about 60% men seem not to want to wash their hands after they finish using the bathroom, where women, about 40% of women don't wash their hands. So it's almost inverted. 40% men wash their hands, and 60% women wash their hands after using the facilities. But the thing is, the chance of transmission of that disease is greatly higher. Also, if you have an infection along with like some sort of Klebsiella pneumoniae or pneumonia, then coughing and expelling various gases or phlegm and that sort of thing. If you've ever seen a sneeze, sort of with a light source behind, it can travel 15 feet. It's disgusting. And when I hear someone sneeze without covering it, it's almost, it's not good. Let's just leave it at that. So, and some bacteria can survive, and some viruses can survive these sorts of things. And I've heard ad nauseum how good the new virus that we have here, this coronavirus, stays on surfaces 3-4 weeks. No, it's exaggerating. 14 days I've seen. But some viruses just aren't susceptible to desiccation. But that's usually the criteria. Even bacteria, they can stay on surfaces for a period of time to be viable. So a lot of the ones that we recover from surfaces, obviously they are viable and they're still living because we can culture them. And just something to think about. And so we can use these. And obviously sexually transmitted diseases are transmitted by obvious means to do that. And food, leaving bacteria on food, let's say the percentage that I rattled off here of 60% men that don't wash their hands, prepare the sandwiches that you buy, or McDonald's hamburger, and they make it and put that, and then you get that and it's sitting on your hands, and you're making the whatever, you get that and people ingest it, and then they can get food poisoning. Now you can get food poisoning by leaving food that was prepared okay, but just being in the environment, let's say Staphylococcus is growing on it, they divide every 20 minutes. So they can produce a preformed toxin that can make you really regret ever eating that potato salad or something. 4 hours later, you'll be having diarrhea and really wish you hadn't had anything to eat. And if it's 6 to 8 hours and you have vomiting and diarrhea with blood, most likely it's salmonella poisoning, which is largely from not washing your hands. Then you probably need to go see a doctor because it's serious. Those can go into your blood. But the idea, we call the ability to spread bacteria or transmit it, is epidemics. Epidemiology is a study of that. And you want to understand the transmission. So this person touches this person, then that person sends that to their, boom, 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 and trying to figure out all that. And same with sexually transmitted diseases, all those sorts of things. So a lot of times you've been hearing about patient zero. It's the initial person that now interacted and started to seed or transmit the disease agent, and then each of these individuals now spread it. And this is how these things work. And I wish it didn't, but that's why we're doing the isolation and things that we're doing now. to stop this sort of thing. And viruses are usually self-limiting diseases. You don't hear that on TV, but that's usually what happens when a virus is formed, and it does its thing. Over time, when it's being a pirate and taking over hosts and things, its genetic information is being modified, and usually it's a perfect state when they start the disease, and over time they mutate to less perfect. That's the philosophy, and that's why I say they're self-limiting. They mutate to a point where they just lose infectivity. This virus seems to be a little bit different that we're dealing with. I don't know. I'm not sure. Do not quote me on it. I am not an expert in that area of virology, and especially with the coronaviruses. That's a different beast in itself, and it's an RNA virus, and it's a nasty one. So anyhow, so we do the disease transmission. I just want to get your comments. I've talked about that. Unit 8 review questions. Pretty straightforward. We've always done these, and I'll give you credit for that. You can just turn in this one page if you want your answers. I did it in Word format, so you can just cut and paste that last page out and put your answers in and save it and email it. I find emailing is most effective. I used to do some of the ways of turning in work on Blackboard, and I've always get a certain percentage of students that for whatever reason the file doesn't transmit through a Blackboard system or whatever. I don't have the time to sort that out, so this is the best. Sometimes students don't put their names on the papers. Well, the email has your name, and I just found that for this particular incident that we had, the email seems to work. So you guys have done a great job, and grading it has been a challenge, but that's my problem, not yours. So I hope this has been helpful. Again, please respond to those polls that I've sent around, especially the one for planning a Lab Practical 2 review. If you want me to go over, if you have questions about any part of the Lab Practical. Now remember, the Lab Practical is really only the chapters that we dealt with from Unit 8 on down. And in some sections the mitosis and meiosis was covered, and to be for consistency's sake, we're going to go ahead for my labs, including the lab only class that I have, is that we'll just deal with 8, 9, 10, 11, and 12, and those will be on the Lab Practical. And so ecology, evolution, genetics, the crime scene analysis, and the microorganisms. And you can obtain the information. The results from the crime scene are been placed on Blackboard. You'll have those, but the review kind of goes over some typified results. Everything should be in the study guide that you need in the lab manual. So I can't count on my students always having the lab manual with them after the event that we're dealing with. So, but I try to reproduce whatever we can. So it'll be pretty much the way we handled before. It is not a comprehensive Lab Practical. It's only these units, and please review that. All right. Well, I'll leave you at this point in time. Stay safe, and please fill out the polls. That helps me for the role, and also getting some feedback as to for scheduling. And we'll get you through this. We're almost there, and now we're going to try to do certain things and activities that help you perform well, and it's all part of the deal. So take care. I'll see you.